Hello, everybody. Today I'll be giving you the lecture uh, for class via this online video. Unfortunately, I've been delayed in Florida and won't be back until after class tomorrow. So instead of canceling class, I'm going to give you the lecture here, and then hopefully I'll be able to answer any questions you might have on this Thursday in class. Also, just a reminder, we won't be meeting Thursday in our normal classroom. I'll send out a message telling you what classroom we'll be meeting in, but it will be in the library. All right, so today we're going to continue talking about comparative genomics, generally, broadly, and we're going to talk a little bit more about some places where we might be applying concepts related to a comparative perspective, and particularly in a genome context. We're going to be talking about some of the main questions that we've started to delve into in the field of comparative genomics, as well as some of the main mechanisms that we think are driving how we do comparisons and ask questions in comparative genomics. So we're going to look at ideas around species concepts today. We're going to talk about the distribution of genome size and the number of genes and some of the odd discontinuities there. We'll also look at some of the distinguishing characteristics for different types of genomes, as well as the role of diversifying mechanisms like gene duplication in genome evolution. And finally, we're going to try to understand what some of the mechanisms of genome change are that we observe at various levels of investigation. So just understand why we are so interested in comparative genomics, just to reiterate that. So recently among the biology faculty, we've actually been looking quite a bit on what are some of the major learning outcomes for our major. And right now, I think there's like 8 or 9 of them. And we've tried to distill them down to about 3 or 4. And one of the major ones is this new learning outcome, which states all biology graduates can synthesize and connect knowledge, skills, and experiences across natural science disciplines, allowing them to address complex questions in the unity and diversity of life. And that's really what this comparative genomics lecture is about, is how can we use genomes to better understand the unity and diversity of life. And just to get us back to some of the core questions that I've talked about previously in this class, a lot of this comes down to how we define what is similar and what is different when we're doing a comparison between species. And what do those similarities and differences tell us about those organisms? We are also looking at the processes that are creating these patterns of similarities and differences. And if we can understand those processes, perhaps we can better understand the organisms we're looking at today. So a general question, and the question that we've asked for a long time in biology, is what is a species? There's a bunch of different definitions for what is a species. You guys might have heard of some of these different species concepts. One of the big ones is the ecological species concept. And this is an idea where species are considered to be groups or populations that share the exact same ecological niche or set of niches. A very popular species concept, seen as one of the dominant species concepts for a lot of the early years of biology. Another species concept that was also popular in early biology and was kind of at the other end of the spectrum of the ecological species concept was the idea of phonetic species concepts. So phonetic species concepts grouped organisms by some sort of similar defining trait. And this was a trait that was typically chosen by taxonomists. So in the ecological species concepts, we have species grouped by the perceptions of ecologists, basically, and phoneticists were grouped by physiologists, developmental biologists, things like that, people who saw similar traits among different organisms. Finally, there was another group in evolutionary biology referred to as the phylogenetic species concepts, And this was a concept where they looked at as a species as the smallest set of lineages or populations that can be recognized by a unique combination of different traits. Now, nowadays we use genetics to determine these traits, but in the past they were actually traits. These were physical observable characters. And we were looking at the smallest set of lineages or populations that could define a unique combination of these different traits. Finally, what came about through the work of Ernst Mayr was the idea of a biological species concept. And this drew a distinct line with what are species in terms of what are groups of interbreeding natural populations that are reproductively isolated from other such groups. So in this case, species had to be biologically viable with each other, be able to reproduce, and be isolated from each other. So it's actually a bit of a combination of some different biological criteria, the reproductive isolation of groups, as well as allowing for some of these ecological species ideas where you could have two populations that are just simply incredibly isolated from each other and might constitute different species. So, what are some of the things that happened when we started delving into species concepts? Well, when we started looking into morphology and the stratigraphy versus molecular processes, we noticed that both are based on inherited traits, right? So molecular processes, DNA, is based on the idea that you've inherited a specific trait. These morphological perspectives were based more on these ideas too, that if I observed a trait, it would also be based on a pattern of inheritance. What instead was found was that often a lot of these quote-unquote inherited traits were not actually inherited by the same mechanism. And with the advent of molecular data, people found that they believe molecular data to be a much better measure of what is an inherited trait as opposed to what is a phenotypic trait that is observable at the level of phenotype. Because you can get to the same phenotype via different mechanisms. What has sort of become the standard in terms of defining species has been an approach known as DNA barcoding. And DNA barcoding is typically based off of using cytochrome C oxidase subunit 1 from the mitochondrial region of an organism, about a 648 base pair segment, as a means of establishing a specific code associated with a specific organism. So in this case, what we tend to see here is that there's a lot less variation within species than between species in this DNA sequence. And so we can use these patterns of variation to say, okay, this is a marker, this is a DNA barcode that we know that is specific to this taxon, and we can use that to identify this taxon. There is a barcode of life database that has all of these barcodes in it. There's over 6 million barcodes in there now. Quite a few, over almost 200,000 animal species, about 70,000 plant species, and about another 20,000 fungal and other species in this database. So it's quite extensive, again, largely based off just this one marker. Right? So, does having a different cytochrome C oxidized subunit 1 mitochondrial region make you a species? That's still, I think, a point that we could argue quite a bit about, because cytochrome C oxidase subunit 1 mitochondrial region has very little to do with anything else that goes on with the biology of organisms, right? It's just a marker by proxy. And you can see that in some organisms, say for instance the rockfish that Cheryl Logan works on, this barcode is not particularly helpful. So yeah, it's typically a good marker, but it is far from perfect. And one of the reasons why is it doesn't actually represent any of the underlying true biology of the species that we might associate with a lot of those other definitions. It holds well to the phylogenetic concepts and these ideas of shared traits, but not so much along identifying what are the biological mechanisms at play. But let's think a little bit more broadly. How would we define a species in a different way from a comparative genomics standpoint? Well, typically, instead of these phenotypic traits now, we often use protein, RNA, and DNA as the standards for how we define species. And there have been some major results that have come about from this process. Probably the first one, and one of the most interesting ones, is that all life on Earth has enough general similarity to show evidence of common origin, right? The general same building blocks are used among all organisms in terms of RNA, DNA, and proteins, that we think that everything basically came from the same place. If we think about the relationships among species, they are typically defined and presented as trees. So here's an example of a phylogenetic tree, showing off the relationships among a wide variety of organisms. And here you can see, we've got mouse and human up here. They're closely related to each other, so they are on the same branch here, and they're in the same what we would refer to as a clade. You'll notice that all vertebrates are in the same clade, so they are all more closely related to each other than, say, the Eukaryotes or the sea squirts, right? Just as all the arthropods are more closely related to each other than they are to any of the chordates or Eukaryotes. Similarly, we have all the fungi on the same branch of the tree, more closely clustered together than any of the animal taxon, and the plant taxon on their own branch down here, forming this clade down here. A lot of these photosynthetic organisms. Using these types of trees, we can break out, using molecular markers, we can break these groups out into large groups that we can easily identify, such as the deuterostomia and the protostomia. And you can see here the number of species that we would associate with each of these different groups, right? With arthropods being the most obvious, high, with the highest number of species. Excuse me. And based upon these differences, you'll notice that there is even our ability to sort of date these trees, to look to say, okay, if we look at these two major groups, the chordates and the echinodermata, when did this group of deuterostomes break away from the protostomes? Where did these two groups break away from each other? And we can actually set a date around 700 million years ago as being the basis for this difference. So that's a typical way that we would present and look at that data, right? And using molecular data as sort of as sort of the driving force for how we draw those trees. Now that's just one example of how we might apply molecular data to trees. There have been a lot of other things that molecular data has shown us. One of the major findings, and this comes from the work of Carl Woese, is that if we look at all living things, they can generally be divided up into three major groups. Here we have the bacteria, the archaea, and the eukarya. And all of this work is based off of looking at 16S ribosomal RNA sequence. And if we look at this sequence, we are able to draw similarities and draw this tree among these groups and show that eukarya have sequences that are more similar to each other than those of than archaea or bacteria. And bacteria have 16S sequences that are more similar to each other than they are to any archaea. In addition to grouping individuals, as I noted previously, we are actually able to use DNA sequence to date historical events. And we do this by using something called a molecular clock. So if mutation is considered to be random in time, we assume that it happens at a regular rate. And if we know, we can count up how many differences we see between two species, and we have some idea based upon fossil record or based upon some other geological signature of when these two species might have had a common ancestor. We can actually say, well, this many mutations is equivalent to this many millions of years. And by doing that, we're able to back calculate the molecular clock of a tree. This is not an easy process. It depends a lot on how mutation works in the system. And so I will say that this takes quite a lot of effort to validate how these molecular clocks are working, but they can be quite persuasive and informative in various instances. Finally, another major result is that horizontal transfer, the movement of genes between taxa, not by vertical transfer, not by passing genes on to your offspring, but by passing genes on to other organisms and species that co-occur with you, has actually become a very important component of how species are created. If we look at, especially if we look at bacterial and archaean lineages, um, we can look at examples where we have species like E. coli, where about 25% of their genes, a quarter of their genes, were actually acquired from other species. What's interesting here is that if we look among all kinds of organisms, we can see massive examples where horizontal gene transfer has been incredibly important. We have seen examples where a Methanococcus species has genes from yeast. We have seen human genes get picked up by tuberculosis. We've seen examples here of Bradyrhizobinium japonicum, where it has two glutamine synthase genes, one that comes from its bacterial lineage, another one that comes from a plant lineage that it's associated with. So we can see all kinds of cases where genes are actually moving between organisms at a rate that's much more frequent than we would have expected. And there's a new book out called The Tangled Tree, which actually talks about this in quite a lot of detail. So you might wonder, if our previous example was a tree, we may be looking at something more like a shrub, right? Where branches are kind of crossing over each other in a way that we previously might not have give them enough attribution for. We can even see examples like this sea slug over here on the right, where they are actually able to eat sea lettuce and uptake chloroplasts, uptake a whole organelle from an organism, and use all those genes that are in that organelle to do biological processes that are very important to it. One of the overriding characteristics of this, though, is that it looks to be that horizontal gene transfer is a lot more prevalent in what are known as operational or housekeeping genes, genes that create something, genes that are involved in biosynthesis. And then informational genes, genes involved in transcription, translation, genes like 16S rRNA, are a lot less likely to be horizontally gene transferred and very rarely do so. So when we look at genomes in general, we might think of them as sort of this global organism now, right? Like when we look at all life on this earth, if we're actually transferring genes back and forth, where do we even draw the lines between species? What has become a species? How is it? How do we apply our phylogenetic species concepts? How do we apply our biological species concepts? When species are intermingling their DNA all over the place, well, what we do find is that when we look at these informational genes, we think that might be the sort of delimiting part of this process, because it's those informational genes that seem to be a lot less subject to horizontal gene transfer, and so may better help us track the genetic relationships among organisms. So when we think about which of these species concepts are actually true and accurate, you can see how genomics kind of pokes a hole in a lot of these and says, yeah, these all may be interested, but even if we look at the shared traits and the way that things are able to isolate themselves, their genes may be moving back and forth at rates that were much higher than what we would have thought of before. However, if these informational genes are distinctly specific from the operational genes, perhaps that is the process that actually drives and helps us define what is a species. So that is a great example where once we've looked at genomes and we started comparing genomes, we notice that a lot of our traditional methodologies to understanding a very important question, something like the what is a species, becomes a lot more complicated when we actually look at what is in the genome of these organisms. When we're not just looking at one gene, like mitochrome cytochrome oxidase, but we're actually looking at a wide range of genes across the entire genome, the picture becomes much more muddy. And it becomes important that we understand how we use patterns and dissimilarity then to understand the processes that we might be driving the relationship in the evolution of species. Here, we can use genomes to help us understand the evolution and the functional relationships among organisms and within organisms. And one thing that we know is that genomes are a record of the evolutionary history of an organism, especially we can look at them in comparison to each other. We can look at the the patterns of shared and dissimilar genetic components to try to understand the progression at which and the time at which various molecular components and hopefully phenotypic traits that we can associate with those molecular components arose in various species. In order to do so, we have to look at a bunch of different types of information, though. We can't just look at one DNA sequence. So generally, we're looking at sort of 4 different types of data. 1, genome sequences. 2, the RNA and protein expression patterns associated with an organism in various contexts. 3, the spatial organization of individual macromolecules, complexes, organelles, cells, tissues, bodies, etc. And 4, what regulatory network, structural arrangements, and adaptive control systems are operating and functional when we look and compare 2 different sets of organisms or more than 2 organisms. So these are the kinds of data we're looking at. So let's use these, let's start looking at some interesting questions as we start to look at these types of data in and among genomes and think about what they tell us about genomes. So one of the things that people look at a lot for genomes is how, what can genomes tell us about the complexity of life, about the complexity of organisms. And one of the things that you generally see is that you see a pattern of correlation between genome size and how complex an organism is. So do we see a correlation between size and complexity here? There certainly is, right? Viruses, some of the least complex organisms, have the smallest genomes, right? And as we go up in what we consider to be complex organisms, those with compartmentalized cells, those with complex life cycles, things of that sort, we notice that we get more and more complex. However, what you will notice is that there are organisms that can have very large genomes, which may not necessarily be overly complex. For instance, if we look at an amoeba, it has a genome approximately 200 times larger than the human genome. And if we look at the lungfish, it has a genome of approximately 43 times larger than the human genome. You could make the argument that both of those organisms are more complex than us, but I think you would have a hard time doing so. So there's obviously some discontinuities there in that argument. So why the different sizes? We've talked a lot about what might drive different sizes in genomes. Particularly, we've talked about the role of repetitive elements or junk DNA. And traditionally, this was referred to as junk DNA. But as we've talked a lot about in class, it was basically called junk DNA because we didn't know if it had any function. What we now know is that the majority of this junk DNA is not transcriptionally inert. It is actually transcribed. Now, whether or not it's functional after it's transcribed remains a point of contention, one of active investigation. But what we have noted is that a lot of it is functionally active, right? So it could be that variation in size has some relationship to our ability to promote transcriptional diversity. Maybe, maybe not. Difference in sizes also would lead to different rates of recombination. Obviously, the larger the genome, the more recombination could have an effect on genome structure, and this could be both a good and bad thing. You may not want to allow for a lot of recombination to occur because it could be deleterious in the long run. What about, so there's gene size, but what about number of genes? Do we see a correlation there? So if we're going to think about complexity, like the more genes you have, the more complex you are, you probably need to have more genes, correct? We would generally assume so. So if we looked across a range of different organisms, E. coli, yeast, pufferfish, plant, Arabidopsis, and humans, we can see that there is definitely an increase in gene density as genome size, decrease in gene density as genome size increases. And we do see an increase in approximate number of genes in these genomes, but you notice that it kind of platforms after a certain period of time. And we don't start getting a ton more genes despite really large increases in the size of genomes. There are a variety of reasons why this might occur. But just because we aren't increasing in the amount of coding sequence in the genome doesn't mean we're not also still increasing in the number of protein products. So here, processes like alternative splicing of genes, where we're using different exons and different combinations to create different types of proteins, may allow for a greater diversity of gene products, even though you only have a consistent number of genes. It's important to recognize the fact that there are different types of eukaryotic, that genomes are organized in different ways in eukaryotes. So there are basically two types of genomes. There's an organelle or cytoplasmic, there are organelle or cytoplasmic genomes, and these are genomes associated with mitochondria or chloroplasts. And then there's the nuclear genome. Main differences here, the organelle genomes are short, the nuclear genomes are long. The organelle genomes are circular. The nuclear genomes are chromosomal. And if they're circular, that means that they must originate from what? What else has a circular genome? Well, if you don't know, you should look up Lynn Margulis and see about her endosymbiont theory about where mitochondrial genomes come from. So I'll just leave it at that. You can research that on your own and figure that out. The other thing about these genomes is you'll notice that for the circular genomes, the numbers are variable. So some cells have more mitochondria than other cells, and some cells have more chloroplasts than other cells. So for instance, muscle cells have a lot more mitochondria than fat cells. Whereas the nuclear genome is pretty much constant among all cells. There's only 2 copies of that nuclear genome in each cell. 2N, in other words, one copy of each set of chromosomes. Also, another main difference between these two types of genomes is that there are no introns generally in the organelle genomes. Not always the case, but largely the case, where there's lots of introns in the nuclear genomes of eukaryotes. And over here we have a diagram showing basically how a mitochondrial genome is set up. A human mitochondrial genome is about 16,600 base pairs long. It's got 22 tRNA encoding genes and 13 protein coding genes, and this control region up here. And this is pretty standard for most eukaryotic genomes. There is variability among plants and fungi, though, in how these mitochondrial genes look. So that's generally an idea of when we think about genomes, what are some of the questions associated with it? What are some of the odd ways that genomes, um, that when we look at some broad ways for defining genomes, how they might tell us some sort of conflicting questions about species, about complexity. Um, so it's important to think about, okay, we've got these genomes that are telling us about all this evolutionary history, but they seem to be telling us conflicting things. So it becomes really important that when we think about how we ask questions from a genomic perspective, that we really think carefully about the level at which we're investigating the genome and what the implications of the investigation at that level are allow us to what conclusions of that level allows us to draw um, about those organisms and those comparisons. So there's basically four ways that we would look at a genome and how it differs. One, we could look at individual bases. Two, we could look at the genes that we see that differ between genomes. Three, we could look at blocks of DNA, large sections of DNA, and say, how do these things differ? And four, we could look at the whole genome. We could look at everything all together and say, well, how do these things differ? So if we think about how these things might differ, we can go first and look at processes like how they differ at the individual base. Um, we talked about this before in some of our other examples, but here we might look at how one specific gene might tell us, or one specific segment of DNA, about what it might tell us about the differences between these organisms. So this is a case where we're looking at a thioredoxin, um, and these are genes that are used to catalyze disulfide exchange reactions. And what we're looking at here are sequences of thioredoxins from vertebrates, human and chicken, from Neospora crassa, which is a fungus, and from a bacterium Staphylococcus aureus. In which you can see when we look at this at a protein level that there are a lot of genes, a lot of proteins that are similar for these thioredoxins, and a lot that are different. If we also look at the genes, at the DNA sequence that underlines these sequence, what we can see here is every time it has a star, this is an identical sequence. Every time it has a colon, this means that you have the same two bases in the vertebrate taxa. And every time there's a space, that means that there's a difference between, there is not a shared sequence between the two vertebrate species. So we can see that every time there's basically a symbol, these are sequences that show conservation between human and chicken, the two vertebrates. And you'll see that there's a lot of these sites that show conservation there, right? And there's large sites and large sections where you might see no conservation with bacteria. There are, however, some sites where there's quite a bit of conservation with bacteria. So in these cases, all three of these organisms need to actually catalyze disulfide exchange reactions. And so they all need a thyrodoxin. But those thyrodoxins of chicken and human are a lot more similar to each other because they're connected more closely on evolutionary time. They've diverged a lot more recent from each other than the bacteria have. The bacteria have been allowed to mutate and change over time, whereas the chicken and the human have not had as much time to diverge from each other. So that's how we might use individual-based variation to look at these patterns and look at specific differences between these groups and describe, which of these groups are more closely related to each other and why. And once we know about their similarities in sequence, we might also draw some inference about how human and chicken thyrodoxins are working in similar functional capacities that may be very different from Neospora and Staphylococcus. If we're going to look at variation at the gene level, one of the major ways that we do this is to look at duplication. So if I'm talking about duplication, I'm generally talking about a specific type of ortholog, and you should all know what type of ortholog is created when a gene duplicates. And I'm not going to tell you, but you got to look it up. So Susumu Ono is a very famous Japanese evolutionary biologist, and wrote a book which has really stood the test of time called Evolution by Gene Duplication. And Ono became well known for his work in this group, in this book. And one of the things that he posited as the hypothesis in this book, well before genetics had really taken off as a field, is that, especially molecular genetics, is that duplication followed by divergence could be an important source of protein novel function. What we found in that in that process is that if you do have duplication, you do have to think about what is going to be the resulting fate of these two copies. How does this function arise? And how can we distinguish what is functionally important and what is just the result of neutral processes? So these are all questions that came about basically through the work of Ono, and people have been invoking his work ever since. So it leads us to the idea, so if you have a duplication, if we look among two organisms, we see that there's differences in the number of these genes. What can happen to that gene from then on? Well, that gene may pass on to descendants, and it could accumulate mutations which may be beneficial or detrimental, or it could just drift neutrally. Or it may not change. It could not accumulate mutations. That gene could be lost altogether. It may no longer persist through time. It may not pass on to descendants. If it's not important, why keep it? That gene may be duplicated in future generations. Perhaps that gene is transferred to another organism. That could also happen. If it's just a duplicate and you already have a copy, maybe you can pass on that other copy to somebody else. Or that gene may fuse or break off a piece or be rearranged into some other context in the genome, whereas that copy can go and do whatever, but that original copy will stay and still preserve its own function. So basically what you have is a case, whereas if you have a duplication event, you have a copy that has to still do what it was doing before the duplication event. But you have another copy that could go down any number of pathways to be either being a functional component or not being a functional component. If we think of a good example of this, we can look at patterns of duplication and dispersal of globin genes. And so what you can see here is this is showing you various types of globin genes and their dispersal across various chromosomes. And what you can see first off is that globin genes are dispersed across multiple chromosomes in your genome. Right? They're dispersed across at least five chromosomes shown in this example, right? And each of these globins has very different and distinct functions, right? So you have an ancestral globin sequence which dates back, more than 1 billion years ago. And that globin has diversified to function as a neuroglobin, globins that are cellular globins. So you've got myoglobins and hemoglobins, and then you've got both alpha chains and beta chains hemoglobins, right? So there's a wide range of these globin genes, and what you not only see is that even within these globin families, if we look at the alphas and the betas, we see that there's also been patterns of duplication here. And one of the things that we know is that as there's been duplication events, these different globins have taken on different roles and different functional purposes. Now, one of the ways that we look and we see whether or not we think, you have examples in these globin cases where we would look at different globins in different species, or we could look among these different paralogs, and we could see, especially when we look in a comparative sense and we say, look at the globins of horse versus the globins of human, we could look and say, do we see how many patterns of non-synonymous mutations do we see in the DNA sequence versus synonymous mutations? We talked about this in class before, but can we match up these terms? Can we match up, if this non-synonymous mutation is a lot bigger than the synonymous mutation? Um, does that mean that it's neutral evolution, purifying selection, or positive selection which is driving this process? If they're approximately equal, are we looking at a neutral, purifying, or positive selection? And if we have a lot more synonymous mutations than we see non-synonymous mutations, are we then looking at neutral, purifying, or positive selection? You should be able to answer that, by the way. That's something that you guys should be able to tell me. But we can look at these examples, and we can look at genes that have the same evolutionary history. We can look at myoglobins among a bunch of different taxa and calculate these values, and then try to figure out what happened. What's the best way to explain what happened to these genes through time? Were they drifting neutrally, or were they acquiring mutations? Are they neutral? Are they purifying? Are they positive? And using these non-synonymous synonymous mutations, we can try to explain those processes. What's noticeable about the globins, obviously, is that a lot of them were retained. So there's been a fair amount of duplication at various points, right? We've got duplication events here, and here, and here, and here. They're all over this tree. So this family of genes was obviously very useful for a wide range of functions. Even if we just look at beta globin at the locus in selected vertebrates, what we can see, is that ancestrally, if we look back before placentals, we can basically see that there are only a few types of globin genes. Specifically, if we look at zebrafish and xenopus, we see that there's only alpha and beta globins back in these kinds of organisms. And this is actually looking at a segment. This is looking at the beta globin locus in all of these vertebrates. So this is the organization of the locus. And what you'll see is that, there's these alpha and beta globins. They're organized in a similar fashion. But as we get into the placental mammals, we'll see that there's a much more diverse set of these beta globins. And interestingly, you can see, there are some taxa that are very similar to each other, like sheep and goat. All right, these are closely related organisms. They have patterns in their beta globin, and their genes are organized in very similar ways. Whereas we were to look at mouse and rat, you'll see actually there's really a big divergence in how their beta globin locus is organized. So even among closely related species, sometimes these patterns are not altogether clear. What's also interesting in this group is that the organization and the patterns of duplications that we see help us understand some of the functional innovations that have occurred back in organisms. So if we look at early placental organisms, we can see that there were basically 5 ancestral beta globins, and these red ones are embryonically expressed, and these green ones are post-embryonically expressed. What we can see is that early in the hominid history, branch of the tree, the prosimian part of the tree, we see that one of these genes, this gamma right here, is actually pseudonized. So that means that it loses its function. It becomes a pseudogene. It can no longer code for a protein. Later, we see a divergence in the New World monkeys from prosimians and Old World monkey, and where both the ancestor here to New World and Old World monkeys has a duplication of this gamma globin. Excuse me. And that gamma globin is actually fetally expressed. So we see additional complexity coming about at this part of the tree. So again, these patterns of gene duplications can tell us a lot about the relationships between taxon and perhaps stuff about the differences that we see in taxon um, and some of the major innovations that might have happened in the history of these taxon. So that's the idea of looking at individual genes, individual bases in genes. But what if we start looking at larger sections? So in that last section, we could see we started looking at all of the beta globins. We started looking at a section of DNA, at a block of DNA. And that's another way that we can think of looking at the level of how things might vary in genomes is by looking at blocks of DNA. Over here on the left, we can see how if we do a comparison between two genomes, we can see sort of if we look between closely related species, all of their genes are organized and oriented in the same fashion, whereas we do a comparison between two different species, we can see that the genes that were organized in a specific way in the previous example, we now have an inversion where this part of the genome has been rearranged and ends up pointing in the opposite direction. Right, so something happened in that genome, a recombination event more than likely, that caused there to be a major inversion in this genome. In this case, we're looking at patterns of what we refer to as synteny, and synteny means same band. So here we're looking at the co-localization of genomes on the same chromosome. And we're also looking for the conservation of blocks of order. So usually they're genes, but they can be other entities, along two sets of chromosomes. So here, we still have the same blocks of order. It's just that they're in a different orientation than we see up here. So this is kind of the way that we think about larger scale duplications. And here again, this is a way for us to look at multi-gene duplications or large rearrangements. So over here we have on the left side of the screen, what you see are some representations of a few different plants. So this is C. rubella, and then two different lineages of Arabidopsis lyrata and Arabidopsis thaliana. And what we see here is that there are sort of these ancestral chromosomes here associated with the C. rubella genome. A, B, C represent ancestral chromosomes, D and E, F, G, and H, I, J, K, L, M, N. All these letters represent ancestral chromosomes and how they've been sort of combined to be on the C. rubella genome. What we then find, though, is that if we look at if we compare the genomes of these two species, we can see that C. rubella and Arabidopsis lyrata, look really similar to each other and show very similar patterns in the grouping of these ancestral chromosomes on the same chromosome. So for instance, if we look at CR2, right, we're going to see it has the ancestral chromosome D and E, and both of those chromosomes are found, those ancestral chromosomes are found on the C. rubella chromosome 2 and also on the Arabidopsis lyrata chromosome AL2. What you will notice is that a little bit of piece of this CR2, though, has located over here to AL1. Another thing that we could notice is that there is a small duplication event here where we see a section of C. rubella 7 going over here to Arabidopsis lyrata 9. Now what we can see there, so these two organisms look to be pretty similar to each other in terms of their synthetic patterns. And they're also strongly showing the relationships of the co-localization of these ancestral chromosomes. However, if we look at Arabidopsis thaliana, what we suddenly notice is that there's been a lot more recombination in this case. So what we see here is we see inversions and splitting up of chromosomes where, say, CR3 has ended up on two different chromosomes, AT2 and AT3, where it was all on the same chromosome up here. We see that a large deletion of CR2 in AT1, and we also see that they all all of this chromosome is on AT1, not split up between two chromosomes like up here. We see cases where we have inversions, where this chromosome has been inverted into two different sections, as well as and combined with an inverted section of this chromosome. So what we see here is this is one of the ways that we can start to think about, well, what are relationships of species? How are these things behaving similarly? One of the reasons why this might be interesting is Arabidopsis thaliana is considered to be our model species for plants. But what we notice is that its chromosomal structure is actually very different than a lot of other related taxa, and perhaps very different from the ancestral taxa. So do our functional experiments in Arabidopsis thaliana, are they good representatives overall of what we might expect from plant function? It's one of the ways we might think about this. So finally, let's talk about the idea of genome duplications. We've talked about looking at things at the individual level, at the gene level, at the block level. Now let's think about the whole chromosome. So basically, genome duplications happen when chromosomes replicate, but they don't segregate properly. And so you end up with multiple copies of a genome in a cell as it's trying to replicate into new cells. The way we identify these is that we look for genome-wide occurrences of pairs of homologous genes appearing in the same order. So in other words, do we have whole blocks, whole sets of genes where we find multiple copies of them, pairs of copies of them that appear in the same order? The textbook example of this is looking at Hox clusters. Do we find one set of Hox clusters? Do we find two sets of Hox clusters? If we find two sets of Hox clusters in a gene, that means at some point we think that there's been a genome duplication. The other way that you need to identify these is that you need to use some sort of molecular clock evidence to show that there's equal divergence times in the many pairs of homologs. So if these genes diverged at approximately the same time, they duplicated at the same time, that means that they should have approximately the same amount of evolutionary distance from each other. There could be a little bit of give here and there, but basically we think they've evolving at about the same rate. That's important because we could just have a pattern where there have been a few genes that have been copied multiple times in a genome. When you do a genome duplication, obviously you go from, say, 20,000 genes to having twice that many. But we know that all metazoans basically only have about 20 to 30,000 genes, and there have been lots of examples of genome duplications in history. So what happens to all those extra genes, right? Something has to happen to them. Well, a lot of those processes we talked about earlier of pseudonization, where they acquire mutations, they're lost, they're broken apart by recombination. Something happens. If they're not useful, they're not maintained. Another thing that can happen is that there are certain types of genes that are able to best take advantage of duplication. So when there is a duplication event, these are the genes that are able to allow for a certain building up of specificity and maintain sort of a functional role. And these are typically genes that are related to either signal transduction and transcriptional regulation, so the signaling and turning off of things, as well as neuronal activity and development. And among all things, like metazoans can have genome duplications, but plants are incredibly susceptible to duplications. So here we often see all kinds of plants that are polyploid, where they have multiple sets of entire chromosomes in each cell. I think strawberry is a good case. I think it's got like 5. It's like it's either 4 copies. It's tetraploid or something like that. I don't know. It's way too many copies of a genome in a cell. But there's also autoploidy. This is when you have all the copies just from one parent. There's alloploidy, where you have copies from both parents. And those are different types of polyploids that can happen. Again, what we're looking for in these cases where we're identifying these synthetic regions and these genomes, overall we're looking to see where divergences in sequence correlate with divergences in species. So these are these genomic differences that we're observing at these different levels. We look to associate those with differences that we see in species. So next class that we have, I'll actually talk about a lot of the examples where we try to show that. For instance, I didn't highlight this when I looked at the large block section, but like if you were to look at, we've got we look at humans and chimps, we've got maybe about a little more than 1% difference in coding sequence, but about 2.7% of the genome has been affected by rearrangement of genomes. And that might have a big effect on phenotype as well. So we'll go and dig a little bit more detail into that in class. Remember, our next class will not be held in our current classroom, but in another room, and I'll send out a message through the iLearn site about that. All right, thanks for your attention, and I apologize for not being able to make it to class, but I hope that this lecture was helpful all the same, and we'll take some time to answer your questions about it on Thursday. Thanks.